Mm. So, hello, welcome everyone to the AWM at Pitt Chapter's Student Seminar Series, the first one for this academic year, 2025-26. Thanks everyone for coming. Today we have two speakers. The first speaker will be Sreshta Venkatakrishnan from Brandeis University. Thanks for agreeing to give a talk. She is actually tangentially related to Pitt, our math department, because she is John Rubin, my advisor's former student, Yang Yang's current student. So yeah, it's nice to always have some ties. And today we'll be hearing from her about some math bio, math neuro analysis. So
details all the time, and then we go to the show.
Hi.
hello.
Hi.
Hi, we just started recording.
Can I begin or?
Yes, please. Go
Okay.
for it. Thanks.
Okay, thank you, Anna, for the invite, and I'm happy to talk to you all about a little bit of the work that I've done so far as part of my PhD. Today, I'm going to be talking to you about the effect of neuromodulation, particularly by norepinephrine on respiratory neurons. I'll give you a brief introduction and motivation, more specifically from the bio side of things, and then I'll give you a brief introduction of our model, and then we'll look at effects of norepinephrine on the different kind of bursting neurons, spiking neurons, silent neurons, and I'll end with a brief conclusion. So the pre-Butzinger complex is located in the ventrolateral respiratory column in the brainstem, and it's responsible for the inspiratory phase of respiration. As you can see here, it projects to the hyperglossal nerve, and the respiratory pre-Butzinger network activity occurs slightly, so it's the inspiratory phase, and it's capable of maintaining intrinsic bursts. So two major sources of intrinsic bursting has been identified, one due to a persistent sodium current and the other due to a calcium-activated non-specific cationic current. And depending on these two types of currents, we can group the pre-Butzinger intrinsic bursting neurons into two different types. In panel A here, so cocktail blocks synapses. So what we see here are just individual neurons that are bursting. So they are intrinsic bursting neurons. And so in the presence of riluzole, which is a blocker for the persistent sodium current, these bursts are still active. However, when you apply cadmium, which is a blocker for the CAN current, these bursts are lost. In our model, we call them C-bursting neurons, and they're driven by CAN current. One other blocker for the CAN current is flufenoic acid. Now, in panel B here, we see an intrinsic bursting neuron, which continues to spike in the presence of cadmium, thereby it's cadmium insensitive. But when we add riluzole, the bursts are lost. So these are persistent sodium current dependent bursters, and in our model, we call them N-bursting neurons. There's another subtype, NC bursting neurons, where both the persistent sodium and CAN current mechanisms can coexist. However, these are very hard to isolate experimentally, so we don't go into as much detail with regards to these neurons in our modeling studies. Now, our goal is to look at the effect of neuromodulation. Neuromodulation is something that occurs in the brain constantly, right? Here's an example where we see a spiking neuron in the presence of norepinephrine. It switches to bursting behavior. A bursting neuron on the bottom panel here, in the absence of the serotonin receptor, loses these bursts and it starts spiking. So, sorry, was there a question? Okay. So in this work, we focus mostly on neuromodulation by norepinephrine, and we focus on this paper by VMRE and Ramirez, where they model or where they perform experiments at the single neuron level, so isolated pre-Butzinger neuron, and they study the effects of norepinephrine. So here, an isolated pre-Butzinger complex cadmium insensitive pacemaker, so N-bursting neuron, In the presence of norepinephrine, the burst frequency is increased. Here we have a cadmium-sensitive burster, so a C-bursting neuron. When we apply norepinephrine, it looks like the burst frequency doesn't change, but the burst duration seems to increase. Silent neurons in the presence of norepinephrine, they continue to remain silent. But perhaps what's very interesting is the tonic spiking neurons. Now, in the presence of norepinephrine, you see that tonic spiking neurons switch to a bursting behavior. Further, under flufenamic acid, which is a blocker for the CAN current, these bursts are lost, while the bursts continue in the presence of a persistent sodium current blocker. So this suggests that the bursts induced by norepinephrine depend on the CAN current, right? So there have been existing works modeling the effect of norepinephrine in these pre-Butzinger complex neurons. In respiratory neurons in general, the effect of norepinephrine is excitatory and is mediated by the alpha-1 receptor. Moreover, alpha-1 receptor activation increases the CAN current conductances. So earlier models, like the Park and Rubin model, as well as the Toporikova and Butera model, they model the effect of norepinephrine via increasing the CAN current conductance GCAN. Doing so, they noted an increase in the burst frequency in N bursters, an increase in the burst duration in the C bursters. However, with just an increase in GCAN, the tonic spiking neurons continue to remain tonic spiking. So we need a different mechanism to see the same results that we saw from the experiments, right? So this is where our mathematical model comes in. We model a single pre-Butzinger complex neuron based off of the model proposed by Park and Rubin, the single compartment model for a respiratory pre-Butzinger complex neuron. One major difference is that in their model, the total calcium in the cell was considered fixed. However, we introduce a voltage-gated calcium current, and so the calcium flux in the cell changes, right? V, N, and H are typically considered the voltage subsystem. Calcium, L, and Ca dot are typically considered the calcium subsystem. And H is the gating variable for the persistent sodium current, and L is the gating variable for IP3. IP3 is the gate that manages the flux with the calcium flux within the cell, so between the ER and the intracellular space. Now, voltage talks to the calcium subsystem via the voltage-gated calcium current here, and calcium talks to voltage via the CAN current here. And depending on the different model parameters, which are the conductances due to GNAP, GCAN, GCA, and IP3, we have various activity patterns in the model. So we have quiescent neurons, we have bursting neurons, spiking neurons, even mixed bursting neurons. Although there are two different colors here, and they're both called NC bursting neurons in this particular case. The difference in coloring is to indicate the different mechanisms involved in the bursting here. In E here, you see that the bursts are initiated before the jump up of calcium can occur. In F, you see that, well, it's hard to see from the figure here, but the calcium jump occurs before the burst is initiated. Okay, and now our goal is to model norepinephrine. So we saw that just GCAN was not enough to model the effects of norepinephrine. So we found from a review article by Exton that IP3 can function as intracellular second messengers for alpha-1 adrenergic agonists, which norepinephrine is. So we propose to model norepinephrine via an increase in both GCAN and IP3 in our model. Doing so, we have a tonic spiking neuron, which when we increase both GCAN and IP3, we were able to induce bursts in these neurons. Further, when we block the persistent sodium channel, these bursts continued. When we block the CAN channel, these bursts were lost, which aligns with the experimental results that we saw earlier. Now, on other cell types, in N-bursting neurons, increasing GCAN and IP3 increased the burst frequency. In C-bursting neurons, the burst frequency doesn't seem to change much, but just by the figure here, you see that the burst duration has also not changed. Silent neurons continue to remain silent. In the interest of time, I'm only going to talk about the effects of norepinephrine on end-bursting neurons, but I'm happy to answer more of your questions if you're interested for the other types of other cell types as well. Now, our model evolves on 3 time scales. So we have P, N, and calcium evolving on the fast time scale, H and L evolving on the slow time scale, and C total, the total calcium in the cell, is a super slow variable. So we have the slow manifold or the critical manifold given by F1, F2, F3 equal to 0. The super slow manifold, which is a subset of the critical manifold, it's in addition to F1, F2, F3 equal to 0, it's given by G1, G2 equal to 0. Now, let's focus our attention on end-bursting neurons. These are neurons in this region here. The first thing we notice is the GCA values for the end-bursting neurons is very low. That means that the total calcium coming into the cell is also very low. So in these neurons, the calcium dynamics do not influence the voltage dynamics as much. And so to analyze these neurons, we can look at the voltage subsystem and analyze the behavior there. Now, we want to understand why we see an increase in the burst frequency with an increase in GCAN and IP3. But to do that, let's first understand the burst mechanism for an end-bursting neuron. This curve here, the S-shaped curve, is the projection of the slow manifold to this subspace, VH subspace. Solid is stable, dotted is unstable. The periodic orbit branches shown here are born at a Hopf bifurcation here, and the trajectory initially travels along the stable branch of the slow manifold until it reaches the saddle node bifurcation here, after which a burst is initiated and the solution starts, the trajectory starts spiking between the periodic orbit branches here. The burst is terminated when the trajectory hits the homoclinic bifurcation and the solution jumps back down to the stable branch of the slow manifold. This is one cycle. So from here, the burst frequency can be estimated by how long the solution spends in the silent phase. That's the distance between the 2 bifurcations divided by the speed, the average speed of H in the silent phase. Similarly, the active phase duration can be an estimate for the burst duration, and that's given by the distance between the 2 bifurcations divided by the average speed in the active phase. Okay. Now, what happens as we increase GCAN, right? From the figure here, we see that the right knee for the slow manifold moves further and further away from the H knock line. That means that the speed of H in the silent phase increases, right? So the denominator here is increasing. What happens to the numerator? To study that, we looked at the 2-parameter diagram of GCAN total, which is just GCAN multiplied with the function of calcium. Remember, we can do this because the calcium dynamics don't play a huge role. The calcium values are very small for end-bursting region, right? So with an increase in GCAN, which is just an increase in GCAN total, we see that the distance between the 2 bifurcations decreases. So the numerator is decreasing. Numerator is decreasing, denominator is increasing. So the silent phase duration decreases, which means that the burst frequency is increasing. Now, in the active phase, more of the solution is now spent below the H knock line, which means that the average speed of H in the active phase decreases. So the denominator is decreasing. We know that the numerator is decreasing. These two effects kind of cancel out. And for the most part, the active phase duration remains pretty constant. Now, since calcium doesn't play a direct role in the end-bursting dynamics, IP3 doesn't play a direct role either, right? So looking at the effect of GCAN pretty much sums up the effect of norepinephrine in end-bursting neurons, which we've summarized here in the 2-parameter plots with GCAN and IP3. Here are various activity patterns according to labels A, B, C, D, and E. The first thing that you probably notice, right, as you look at the frequency diagram here, is that the frequency increases in discrete steps. To understand that, we looked at the bifurcation of the full system with respect to GCAN. And so initially, we are here on this stable branch. These three are bursting branches, right? So initially, the solution travels along the stable branch until it hits the saddle node bifurcation here, which corresponds to the threshold value for IP3, equal to for fixed IP3 at that GCAN value, right? So once it hits the saddle node, the stability for that branch is lost. And so with further increase in GCAN, it jumps up to the next stable bursting branch, which is the branch in light blue. So we see frequency in this range here. Then further increase in GCAN, we jump again to the next stable branch. And at the saddle node bifurcation denoted by square here, there's no further bursting branch, stable bursting branch to jump to with further increases in GCAN. So it jumps to the next stable branch, which happens to be the tonic spiking branch, which is why we see here that bursts are lost and the solution starts spiking with further increases in GCAN beyond that.
Quick
We,
question. Oops.
yeah,
You mind, the frequency here is the frequency of the bursting or the spikes within the
it's
burst?
the bursting, the
Okay.
entire, so the entire period, 1 over the entire period.
Okay, thanks.
Yeah. We also observed a very interesting phenomenon here. With each of these thresholds, crossing each of these thresholds results in a loss of one spike within the bursts. We think that's because the burst duration, as you can see from this figure here, remains pretty much constant. So the burst duration remains pretty much constant, but the frequency of spikes within the burst decreases. And so we end up losing one spike per burst as we cross each of those thresholds. Okay, now let's move on to the next interesting case, which is the tonic spiking neurons. These are neurons in the intermediate GCA range, and we want to understand how with an increase in GCAN and IP3, we're able to induce bursts in them, right? So the star here denotes the control case. With just an increase in GCAN, we notice that a tonic spiking neuron continues to remain tonic spiking. Now, what happens with just an increase in IP3? From panel A here, we see that we are able to induce bursts. However, when we block either the persistent sodium channel or the CAN channel, the bursts are lost. This was not what was observed experimentally. Now, what happens when we increase both GCAN and IP3? Again, we're able to induce bursts. Further, when we block the persistent sodium channel, these bursts continue, and when we block the CAN channel, the bursts are lost. This indicates that the bursting is CAN current dependent, which aligns with from the experimental observations before. Now, let's quickly understand the bursting mechanism here. Traditionally, CAN current dependent bursters, in those kinds of bursters, the bursting is driven by calcium oscillations. So calcium jump up triggers a burst initiation in the voltage compartment. However, if you see here, an initiation of burst in voltage occurs before the jump up of calcium. So this suggests that the mechanism here is different. So here you see the surface in violet here. That's the slow manifold projected to the V, calcium, and H space. So the trajectory travels along the stable branch, the lower stable branch, until it hits the fold of the slow manifold, which is the blue line here, projected onto 2D. And so you see that the initiation and termination of the burst actually occur when the trajectory crosses the fold, right? Does this mean that calcium is not essential at all in this case? No. If we did not have a jump up of calcium, jump up and eventual jump back down of calcium, the trajectory would continue spiking. But because a calcium jump is initiated, and which means that calcium returns back, jumps back down, the trajectory crosses the fold again, terminating the burst. So we do need calcium oscillations to maintain these bursts. Now, why is an elevated GCAN necessary for DNA-induced C bursting? We saw that we were able to induce bursts with just an increase in IP3, right? Now, to understand that, we look at, we block the persistent sodium channel completely, and we look at the system, reduced system then. The system has a stable equilibrium point in this case, with just an increased IP3. And so we're not able to induce oscillations, calcium oscillations, right? So the bursts, so we do not see now when we increase both GCAN and IP3, this equilibrium point that was stable now becomes unstable, enabling calcium oscillations. And so we saw that that was necessary to to terminate the bursts, right? And so because we have calcium oscillations, we need both GCAN and IP3 in order to be able to induce the kind of bursts that we saw experimentally. This is just a 2-parameter diagram, GCAN with GCAN and IP3. Notice how we need a high enough value of both GCAN and IP3 in order to induce C bursts, right? The bottom traces the amplitude of calcium oscillations. So you see that with lower GCAN values, the amplitude of calcium oscillations is very low. That results in these subthreshold oscillations. So if for us to have like full length calcium oscillations, we need a higher GCAN value. Okay. In conclusion, in the case of N-bursting neurons, the burst frequency increased, but the burst duration remained pretty similar. That aligns with experiments. C-bursting neurons I have not talked about at all, but we noticed an increase in the burst frequency, either an increase in the burst frequency and duration, or both the burst frequency and duration remained similar. This does not fully agree with experimental observations, but we think it's due to the potential role of the voltage-gated calcium channel that we've introduced. I'd be happy to talk more about this if we have time. Then we looked at the case of tonic spiking neurons, where we were able to induce CAN current-dependent bursting. This also aligns with experiments. And silent neurons continued to remain inactive in the presence of norepinephrine, also aligns with experiments. We have some interesting novel predictions in our model. We noticed the burst frequency in N-bursters increases in discrete thresholds. In also, we noted a loss of spike within each of those N bursts as we cross each of those thresholds. We saw a GCAN-induced increase and IP3-induced non-monotonic changes in C bursting frequency. Again, I've not spoken about these here. And we also saw some complex bursting dynamics in the model. But perhaps the most important discovery was the mechanism by which we're able to induce CAN-dependent bursting. From the point of view of conditional pacemaking, this is very interesting for us. So right now, we're working on extending these single isolated Pre-Butzinger complex neuron results to the network level. We did some preliminary simulations, and we saw an increase in the duration and the frequency of the bursts that aligns with the experimental observations also. However, we noticed a significant decrease in the synchrony at the network level. And so we are trying to incorporate effects of neuromodulation on synapses and extend this to a larger network, because this simulation was with just 15 neurons, right? And this is done in collaboration with a postdoc that's working with Yang right now, Nate. A couple other interesting directions. We are trying to test the involvement of IP3-dependent mechanisms, and also potentially the role of GCA, because that seems to play a huge role in our C bursting dynamics. We're also looking at the mixed bursting and MMBOs in the model. We have a bunch of those, and these seem to occur at the transition of tonic spiking to bursting. So in the middle of when applying norepinephrine, in that transition phase, we observe a lot of these mixed bursting dynamics. And so these dynamics are not just interesting from, like, they're not just mathematically rich, they're also very interesting from the point of view of just looking at the effect of neuromodulation by norepinephrine. Those are mixed bursting, and we also have MMBOs in the model. So I'm currently working on analyzing the mechanism behind these kinds of rich activity patterns. Yeah, with that, I'd like to thank my advisor, our collaborators from U Chicago, and thank you all for listening. I'd be happy to take any questions.
All right, thank you very much.